Cell cytoplasm and cell organelles. Good morning, everyone. Today we will discuss one of the most important topics in cell biology, cell cytoplasm and cell organelles. Every living cell contains different structures called organelles that perform specialized functions essential for life. Just as different departments work together in a factory, each organelle performs a unique task to keep the cell alive and functioning properly. The cytoplasm is the region between the cell membrane and the nucleus. It contains the cytosol, organelles, enzymes, nutrients, and various molecules necessary for metabolism. By the end of this lecture, you will understand the structure of the cytoplasm, identify the major organelles, explain their functions, and relate them to human diseases. Learning objectives. After today's lecture, students should be able to define cytoplasm and cytosol. Describe the composition of cytoplasm. Explain the structure and function of each cell organelle. Compare different organelles according to their roles. Understand how organelles cooperate to maintain cellular homeostasis. Recognize the clinical importance of organelle dysfunction. These objectives provide the foundation for understanding physiology, histology, pathology, genetics, and molecular biology. What is cytoplasm? The cytoplasm is the semi-fluid material located between the plasma membrane and the nucleus. It occupies nearly 70% of the cell volume. It consists of three major components: cytosol, fluid portion; cell organelles; cytoplasmic inclusions. The cytoplasm is the site of numerous biochemical reactions, including glycolysis, protein synthesis, intracellular transport, and signal transduction. It also provides mechanical support to organelles and helps maintain the cell shape. Without cytoplasm, organelles would not be able to communicate or perform their functions efficiently. Cytosol. The cytosol is the liquid component of the cytoplasm. Approximately 70 to 90% of cytosol consists of water. The remaining portion contains dissolved proteins, amino acids, glucose, lipids, electrolytes, enzymes, vitamins, RNA, and metabolic intermediates. Major functions include transport of molecules, medium for enzymatic reactions, storage of nutrients, regulation of intracellular pH, maintenance of osmotic pressure. Many metabolic pathways, including glycolysis, occur entirely within the cytosol. Cytoskeleton. The cytoskeleton is a network of protein fibers extending throughout the cytoplasm. It consists of microfilaments, made of actin protein. They help in cell movement, muscle contraction, cytokinesis, and maintaining cell shape. Intermediate filaments provide tensile strength and structural stability. Microtubules, made of tubulin protein. They form cilia, flagella, centrioles, and mitotic spindle fibers. Functions include maintaining cell shape, supporting organelles, intracellular transport, cell movement, cell division. Ribosomes. Ribosomes are the smallest organelles but are among the most important. Their primary function is protein synthesis. They consist of ribosomal RNA and proteins. There are two types. Free ribosomes produce proteins used inside the cell. Bound ribosomes attached to rough ER and produce proteins for secretion or membrane insertion. Protein synthesis occurs through the process of translation. Thousands of ribosomes may be present in one cell, making them essential for growth and repair. Endoplasmic reticulum (ER). The endoplasmic reticulum is an interconnected membrane system. There are two types. Rough ER contains ribosomes. Functions: protein synthesis, protein folding, protein modification, transport to Golgi apparatus. Highly developed in pancreatic cells and plasma cells. Smooth ER has no ribosomes. Functions: lipid synthesis, steroid hormone production, Drug detoxification. Calcium storage in muscle cells. The smooth ER is abundant in liver and adrenal gland cells. Golgi apparatus. The Golgi apparatus acts as the packaging and shipping center of the cell. Proteins synthesized in the rough ER are transported to the Golgi. The Golgi modifies proteins by glycosylation and phosphorylation. It sorts proteins and packages them into vesicles. Functions include protein processing, formation of lysosomes, secretion of hormones and enzymes, membrane renewal. Without the Golgi apparatus, proteins cannot reach their proper destination. Mitochondria. Mitochondria are known as the powerhouse of the cell because they generate ATP through aerobic respiration. Each mitochondrion has outer membrane, inner membrane, cristae, matrix. Functions include ATP production, cellular respiration, fat metabolism, calcium regulation, initiation of apoptosis. Mitochondria contain their own DNA and ribosomes, supporting the endosymbiotic theory. Cells requiring more energy, such as muscle cells, contain numerous mitochondria. Lysosomes and peroxisomes. Lysosomes contain hydrolytic enzymes. Functions: digest bacteria, break down damaged organelles, recycle cellular waste, destroy foreign particles. Known as the suicide bags of the cell because enzyme leakage can digest the cell itself. Peroxisomes contain oxidative enzymes. Functions: break down fatty acids, detoxify hydrogen peroxide, neutralize toxic substances, support lipid metabolism. Catalase converts hydrogen peroxide into water and oxygen. Centrosome, vesicles and vacuoles. Centrosome contains 2 centrioles. Functions: formation of spindle fibers, cell division, organization of microtubules. Vesicles, small membrane-bound sacs. Functions: transport proteins, transport lipids, cell signaling, exocytosis, endocytosis. Vacuoles, storage organelles, store water, food, waste materials. Plant cells possess a large central vacuole, while animal cells contain small temporary vacuoles. Summary. Speaker notes. Today we studied the cytoplasm and its major organelles. Remember these key points. The cytoplasm is the site of most cellular activities. The cytosol provides the medium for metabolic reactions. Ribosomes synthesize proteins. Endoplasmic reticulum manufactures proteins and lipids. Golgi apparatus modifies and packages proteins. Mitochondria generate ATP. Lysosomes digest waste. Peroxisomes detoxify harmful chemicals. Centrosomes organize cell division. All organelles work together to maintain life and normal cellular function. Understanding these structures is essential for histology, physiology, pathology, microbiology, genetics, and medicine.